So what I'm going to do in the time remaining is talk briefly to give you the background, well maybe not so briefly, for about 45 minutes or so, to give you the background that will help you understand what we're going to be doing in the hands-on session, and in particular why we're going to be doing it. So this is something that is quite new. Alex was talking about developing hybrid expertise. And so people generally who come at HPC come from a computational side looking for an application of relevance, and they will work their way through different things of interest to them and might find themselves looking at biomedical things, medicine, engineering, and science. And from the science side, we are starting to appreciate that the computational power is far more sophisticated than it's ever been previously, and we can start to ask questions that can be addressed computationally that we can't necessarily look at experimentally, and so we're coming from the experimental side. So innovations that we have been doing in training with CompBiomed involve taking one or other population of users and providing them with the ability to develop a dual expertise. It's like speaking a different language, speaking a computational language and speaking an experimental language. So, my background is not at all computational. It's mostly experimental. So, I'm a professor of biochemistry and molecular biology. I am at University College London. Like many of you and many people, I have been pretty much around the world, living in different places. I have dual nationality. I've lived in 4 different countries for at least 4 or 5 years each. And I am particularly interested in understanding about education, because I think education is really important. It's a driver for knowing how you're going to position yourself and how you're going to engage with people and things, for informing yourselves about how you can do that. And I'm extremely interested in integrating research and teaching, because much of the teaching that people think about relates to background information that doesn't look exciting when you necessarily see it in a class, but was Nobel Prize winning 50 years previously. So what I'm interested in doing is bringing some of the current things that we do in research into a teaching environment. And this also gives an advantage of being able to create new disciplines and new interfaces. So these hybrid areas are areas where a lot of change happens very rapidly, has a lot of opportunity, and they're good places to be if you can find yourself one of them. My particular research interests are in personalized medicine and in G-protein coupled receptors and drug discovery, because let's face it, the clock is ticking, I'm not getting any younger, and I want to be able to accurately make the things I'll need when I get older. Thin pills, cognition enhancers, all these sorts of things. This is a purely personal approach to personalized medicine. But more seriously, as we know more about genome sequences, it will become increasingly possible to take account of individual variations. So you will be able to have bespoke clinical treatments based on your particular physiology, your particular genome sequence. This is a very democratic system, and for it to work, everybody needs to be able to, as effectively as possible, integrate computational and experimental methods, because it's expensive. And the computational analyses and the rational design that Alex described, and not just screening every compound you have in the box, is the way in which to save money. So we are looking at integrating methodologies to save money so that you can develop solutions for everybody. So I'm particularly interested in new fields of study, new ways of teaching existing fields, and I think that probably in 5 to 10 years' time, we will see that amongst the specialties in clinical medicine, like orthopedics and gynecology and dermatology and neurology, we'll start to see things that involve looking at molecular medicine and computational medicine that take data and large amounts of data, imaging data, sequence data, structure data that tells you, for example, if you need a pacemaker placed in a particular place, if you need a stent designed of a particular way, if you need some kind of a prosthetic designed, all these sorts of things are going to be up and coming. My background is broadly divided into 4 areas: research and drug discovery, teaching innovations in e-learning, and I teach everything from medical students and undergraduates through to postgraduate students and PhD students. The knowledge transfer is the thing that is very important for all of us, whether we're in industry or in academia. This idea that you pass on what you've learned and you help train other people. So I remember who trained me. You sort of have a family or parents, you have a lineage of your PhD supervisor had a PhD supervisor and another. So you can work out who your grandparents are, how many generations you are from respected and esteemed people. So this is important background to know the context in which you're working. And enabling is things like this. Coming here and being able to talk to a new group of people who might not have seen this before. You may like what I show you, you may not like it, but you'll have a chance to look at it. Turn it around, play with it a little bit, see what you think. So let's start. I'm going to divide my talk into these areas. I'm going to give you a very rough guide to molecular biology, what you need to know to put into context what we'll be doing in the hands-on session so that you understand why we're doing it and why it works. We'll talk about metagenomics, which is a very hot field. You probably have as many bacteria in your body, perhaps, as you have your own human cells. So you're a little ecosystem. And they've made links between populations of bacteria, for example, and obesity, depression, things like that. So there's a great amount of interest in bacterial passengers that we carry with us. And we're going to look in particular at one of the biomarkers that we use for understanding microbial diversity in metagenomics. And then I'll describe for you the experimental methodology that was used to generate the data that you'll be looking at. And we'll be looking at this data in the computational session. So this is a pretty standard molecular flow of DNA makes RNA makes protein. Many people know about these things. Many people know about DNA being a double helix and where your genetic information is stored. And this is where genome information becomes interesting for people. There was a question earlier about what if there's a mutation in a protein and how do you take account of that? Well, it depends on how prevalent it is. You can model these sorts of things. For example, the cystic fibrosis disease, most of the mutations or most of the defects that have been characterized come down to a single amino acid. And that is easily modeled and addressed and targeted on scale, if you like. So DNA is made into RNA. Most people think about the messenger RNA as being the important one because it's the one that is read to be turned into protein. But if you didn't have these other kinds of RNA, the transfer RNA and the ribosomal RNA, which make the translational machinery that reads the code that's in the messenger RNA, you wouldn't have a protein. So these things all work together, and the ribosomal proteins in particular are quite interesting. So your translational machinery is a ribosome. It's basically 2 parts put together, a large part and a small part, and each part is made up of RNA, the ribosomal RNA, and of proteins, the ribosomal proteins. And this just shows you what happens in bacteria, which are prokaryotic, and this shows you what happens in eukaryotes, which are multicellular organisms, mammals, us, yeast, things like that. And you can see that there are ribosomal RNAs, they're indicated with an S here, that go together with proteins to make the large and the small subunits of the ribosome that go together. And we are particularly interested in this species here. In prokaryotes and in bacteria, this is the 16S ribosomal RNA. We don't have it in our human cells. Eukaryotes have an 18S ribosomal RNA. So the 16S ribosomal RNA is unique to prokaryotic species, and we can take advantage of that to do the metagenomic studies that we're going to look at now. So metagenomics is simply the analysis of microbial species in some kind of a sample. It can be anything. It could be soil. We're going to look at some soil microbial samples today, the sequences from them. It could be water. You could do this for pollution studies. You could see what species are most likely to grow in lakes or rivers that have more acid pH or more alkaline pH. It can be your body. You have many different microbiomes in your body. Your skin, you have the skin microbiome that you have from birth. People have a different skin microbiome if they were born through a normal delivery as opposed to a cesarean section. So you get your first microbiome when you're born. You have microbiomes in your mouth. This tells you something about the bacterial species, the gut microbiome, which is of influence on obesity. So they're all over the place. And basically what people do is they purify the genomic DNA from the bacterial species, and they look at the ribosomal genes. In particular, they look at this 16S ribosomal gene that we saw was unique to bacteria, and they use the information in these sequences to characterize the different species of microbes that are in a population. And we'll look at how they do this, but in short, they take and line up all the genome sequence of the 16S ribosomal DNA from different bacterial species in the way that Alex showed with the multiple sequence alignment, and they look for regions that are similar and regions that are different. And in most microbial species, there is a particular hypervariable region that has much variation between species and is flanked by two very conserved regions. So if you use those conserved regions to extract that bit of sequence from your sample, you can look at the hypervariable bits in the middle and see what kind of diversity you have there. And that's basically how these data were generated. You can do a slightly similar thing in eukaryotes if you want. You can use the 18S ribosomal DNA, and there's a region there, but we're looking at bacteria. This is what the 16S ribosomal RNA gene looks like. It has a lot of secondary structure in it that's formed. This is the RNA, and you can have base pairing in DNA. It holds the double helix together. You can also have the same thing in a single-stranded molecule of RNA locally, and these are all the regions. The stems are formed where there's this hybridization, and the loops are in regions where you don't have this base pairing holding it together. And if you take this molecule from one end, here we've got one end here, and the starting end, which I always lose here, there we go, the start end and the end rather, and pull it out in a linear sequence from start to finish and look at the level of similarity, you can see these regions where there's very little similarity, and you can see these regions where there's high levels of similarity. So you can pick DNA sequences to amplify between 2 points, so taking this common sequence and this common sequence and looking at everything in between. And this is, we'll look at it in more depth in a moment. This is a technique that you see on all the forensic shows, polymerase chain reaction, where they make large quantities of DNA between two very precise points. And to do this, we will pick our regions of very low variability, here and here. So the F is forward and the R is reverse. I'll show you why that's important in a moment. And it's the very variable region in between that we want to be looking at. So these are the sequences, the DNA sequences that we're going to be using. These are very specific pieces of DNA that will be annealing or sticking to the microbial DNA in our sample, and we will then be able to amplify the regions in between. Now, this is something that, in order to get a single primer, when you run a multiple sequence alignment, sometimes you have to play tricks. So for example, in this primer, its sequence starts with G, T, G, and this Y here means that in some of the bacterial species in the multiple sequence alignment there was a T, and in some of them there was a C. And this is the nomenclature for having degenerate sequences in your primers. So when this primer is being synthesized, and they're synthesized chemically, at this position, some of the molecules will have a T put into them, and some of the molecules will have a Y put into them. And this is how you do your best to make sure that your primers will recognize every possible conserved region that's out there in the bacteria, so that you have richness in your sample. Polymerase chain reaction is something where you separate the two strands of DNA. You use heat to do that because heat will break those bonds. You allow your specific primers, the F and the R primer, whose sequence we looked at on the previous slide, to anneal. It's a slightly lower temperature that allows them, because they're short and they have a melting temperature, that they need to anneal to the specific sequence of DNA. So if it's too high, they won't bind. And if it's too low, they'll bind everywhere, or more places than you'd like. And then this third step is extension, where you take a polymerase which makes DNA. So you take a DNA polymerase and it fills in the gaps going from where this primer has annealed, it fills in the gaps, and from where this primer has annealed, it fills in the gaps there to make the second strand. So you can see that it's 2 to the n, it's an exponential reaction, and as you go through these cycles of 3 steps, each time you double the amount of material you make. And at that point, you have a large amount of that V4 region of the 16S ribosomal RNA that we were looking at. The primers are the conserved regions, and we want to know what's in the middle. We use a technique called next generation sequencing. I have shown you 2 different examples here of it, and this is something that generates a huge amount of sequence information. It's a tiny little chip, and it will generate about 25 million sequences that come back from this. So you're trying to sequence at sufficient depth to make sure that every unique piece of data that you have in your sample is represented in your sequence. The way in which this works, there are 2 different technologies, but basically this is the primer that has annealed to a template strand, and it will be continuing synthesis to make the new strand of DNA. The polymerase reads what the sequence is on the opposite strand, and it knows what to put in to the new strand at the appropriate place. This particular type of sequencing, this is Illumina sequencing that we've been using. There's also a technology called Roche 454. But what it does is it sits the DNA polymerase there. You can see that this A has a T that needs to be added because you have AT base pairs and GC base pairs. You may have heard of these. When the T is added, it has a terminator put around it, and a flash of light comes in, and it takes a snapshot, and then the terminator is removed, and the polymerase moves on, works out what the next one is. It's also a T, so it puts the T in there, it puts a terminator around it. There is an image taken of what that new nucleotide is, the terminator is removed, and the polymerase goes on. So you go through this stepwise process of putting in the appropriate nucleotide, blocking it so nothing else can be added, taking an image, and then unblocking it and moving on. And if you look at the cell over time, you can see at a specific position, because of the color that's been added, what the sequence is of the DNA. And so you take your 25 million reads and look at your results. Now the primers that we use, so we've looked at the 515Fs, the 515Rs rather. We have a forward primer and a reverse primer. We can put sequences into them to barcode them. So if 5 people did individual samples, we could put a barcode into their primer, give that primer a number, and then they would know from the data that came back from the sequencer which sequences corresponded to a particular sample. And that's how we're able to look for comparisons and find associations in data sets. So if we all took a look at a sample of the microbial species present on our skin, for example, and we had metadata associated with that, our age, our gender. Were we on antibiotics? Did we have a skin condition? Had we been on public transport? Any of those sorts of things. What kind of diet do we normally eat? We might start to be able to see associations between the populations, the diversity in populations and the samples in populations, and some corresponding piece of metadata. Now, we are going to be looking in this hands-on exercise at a combined pool of soil data and skin data. We have stripped out all of the metadata from them. You don't have enough time, unfortunately, in 2 hours to go through all the experimental things and see how to do this. So we're giving you these data, but we're giving you a very large data set because this is the kind of thing that supercomputers are perfect for. For students previously, before we started using HPC in our studies, we used to give them a stripped down data set. But now we actually are using things in the Comp Biomed sequencing HPC facilities, and we can use a full set. So we have stripped out all of the metadata. The only thing you'll be able to identify is soil or skin. And skin means it came from a human microbiome sample. Soil means it came from some London park somewhere that our students will have gone and collected from. And there's some really interesting experiments you can do with that. So we've been having real fun with this. You know, the same part of the park over time. One year we had people who found an awful lot of acid-loving bacteria, and we got them to take a photo of where they'd done their sample, taken their sample from, and there was a rhododendron there. And rhododendrons are acid-loving plants, and so the gardener in the garden had been feeding it with things that kept the soil acidic, which is why it promoted the growth of acidic bacteria. Well, we can pick out where there are that kind of plant, more or less, from the sorts of bacteria we see. So you will be looking at these samples. And this is the experimental workflow that people have gone through to generate these data. So you collect the sample, and it's as important that you collect the metadata that go with the sample, because the metadata are all of the parameters that you have available to you at that time that tell you about the context of the sample. So for our soil samples, we have the time of year, the temperature, we now take the pH of the soil, we have the GPS coordinates, we can go back to the same place year over year and see if there's any variation in that. There's all sorts of experiments you can do as your data set gets larger. You then isolate the genomic DNA from the bacterial samples that you have. We check its quality control because next generation Illumina sequencing is expensive, and you want to make sure that the sample that you put on it is good because, as Alex has said, good data in, good results out. Not so good data in, not so good results that come out. Then we do the polymerase chain reaction with the bar-coded primers, and we check the quality of the PCR amplicon on the gel. This is really important so that the sequence quality that you get back is maximized. And usually that's a number of reads, so there are quality scores on your sequence. And then we purify the amplicon, we quantify it, we pool everything together because we're doing this in a group, so it's not one read of 25 million per sample. We usually have between 40 and 80 samples on one chip, so they get maybe 300,000 to 500,000 sequence reads back. We send it off to the Illumina sequencer, and then we get to the data analysis stage. And the data analysis stage is where you come in and where our hands-on workshop is going to start. I'll just show you really briefly. We've been able to do this protocol over about a 24-hour period. So we had broken it down into 8 sessions of 3 hours each, but you can split it up any way you like. And it depends really on how much resource you have available to in terms of time. Your collection of samples is something that, well, if you have to go collect soil from somewhere, you'll have some transit time to get there and some time to collect and collect the data around it. If you were collecting the microbiome from your skin, that's something that you can do just in the lab just before you go to purify it because it's readily to hand. And we then, because we are teaching medical students who have very limited computer experience and are very familiar with visual interfaces for accessing computational things and much less familiar with the command line. So teaching people how to work with the command line, looking at this program chime that we will be looking at in more detail because this is what you'll be using for your analyses, and then purifying the DNA and then going through how do you use a high performance computer, which again for people who are not familiar with external computational resources other than what they have in their own laptop or in their own phone or in their own iPad, involves figuring out how to log in as a user, navigate your way around, what the file structure looks like, what the path structure looks like. So there's some work that is done there, and then they start to do the data analysis. So these are the two bits that we're going to look at. Chime is what you will be using for analysis. So some weeks ago, we were liaising with the Barcelona Supercomputing Center and the collaborators and the colleagues that we have here through CompBiomed, and they have installed chime on Mare Nostrum for us. We have tested it, and we have got it working. My hope is that you will get it working and get it finished in the time that we have in this workshop. But in case you don't, I also have a sample output file so that you can have a look at how to analyze the outputs that you would have or expect to have back. In general, it should take about an hour and 10 minutes to run the 3 sequential scripts that are needed to go through this. So let's take a look at chime first. Okay. So chime is a program that has been developed for taking a look at microbial ecology. So that could be microbial ecology is chime.org if you want to fire it up and have a look. It could be microbial ecology that is in a pond or in a park or on you, anything like that. And one of the things that's particularly interesting about it is that it was not designed for HPC. It was designed for single standalone use from an individual looking at something on a particular computational resource available to them. So this is one of the interfaces that I was describing to you, and one of the reasons why these areas are both fun and challenging to work in, because you have to make something work in the computational environment that you have available to you. So things that are not porting code that was never designed for HPC onto HPC and figuring out how to make it work and how the batches work and where you save things, what your scratch directories are and things like that has been a real learning process for us. And it's different for each of the HPC facilities that we've been doing this with. So it's quite interesting. And if you are going to be going on and working more with HPC and porting codes or developing codes for them, architectures and things will become quite important to you, and the way in which you structure things will be important. For us, I am hopeful that as more biomedical applications start being developed and as more people start in the academic and the industrial and the clinical side of things start to use these sorts of computational resources, that we might be able to better inform people of the kinds of architectures that would work optimally for the sorts of analyses that we're going to be doing. And here is a second bit. This is the link for the workshop that we're going to be doing today. And this link I also have in a printed out version so that you can make some notes for yourself if you find small typos that are grinding you to a halt, which I'm sure there will be. We've tried our best to strip most of them out. And it explains how you get to Mare Nostrum. I understand that everybody has an account in Mare Nostrum. Have you actually logged on using it? Good to go? Excellent. So we found that when we're doing workshops, the easiest thing to do is make gating points. So the first gating point that we're going to do is get everybody logged on to Mare Nostrum, and then perhaps we'll have coffee or something like that. And then as we go through the different bits and pieces in the workshop sequentially, we'll just stop and make sure that everybody's at the same point and see how quickly the queues are moving and if people are encountering difficulties. So that is essentially what I wanted to present to you in the portion that I was going to be talking about. Before we take a break and start with more of the hands-on workshop bit, do you have any questions about any of this, either in very specific detail or more generally, more broadly? No? All good? Well, if you have questions as we go through, please feel free to ask and we can help sort you out for them. And I think that's the end of what I was going to speak about. We're 15 minutes in advance, so maybe we could take a coffee break now and then start the hands-on thing exactly at 4 o'clock. Would that suit people? Right, okay, excellent. Thank you very much. We're done.